Hey, Marty, you ready to play some biology trivia?
But of course, ask away.
All right, so here's your first question. How many researchers would you guess study the fruit fly, Drosophila melanogaster?
Oh, I know that's a big number. I would guess, 6,000?
Well, not too bad. According to Google, it's over 10,000 researchers spread out over 3,000 labs worldwide that study the fruit fly. All right, let's do another one. How many researchers do you think study the worm, Cenobites elegans, or C. elegans, as it's often referred to?
Oh, man. Probably fewer. Maybe 2,000?
Okay, not bad. Good estimate. Maybe around 4,000 researchers with about 1,600 registered quote unquote worm labs around the world. Okay, one last one. How many researchers do you think study mice?
Oh, man. Mice dominate biomedical research. And there's a lot of infrastructure that's needed to house them and care for them and all sorts of repositories funded by governments. This was tricky. I'm going to say 50,000.
All right, well, I'll confess, it was hard to find a definitive answer to this question, but the estimate is probably closer to 100,000 researchers worldwide. And we also know that there's over 192 million rodents, so that would be mice and rats combined, that are studied each year.
Woo, that's a lot of rodents. Okay, this is fun, but where are you going? What's your point? Are you trying to poke me and get me talking about why I don't like model organisms again?
Okay, sort of. You and Art already discussed some of your concerns about both the term model organism, but also some of the drawbacks for biology of working on only a few number of lab organisms back in episode 87 with Sabina Leonelli and Rachel Ankeny.
Yeah, and maybe if anybody wants to hear me whinge on that topic, they can go and listen to that episode.
Fair enough. And why I bring up model organisms in the first place isn't really to get you to complain about them. It's more to contrast lab organisms to what people might call a field model organism. I mean, take your research, for example. You're part of an international group of researchers who study the house sparrow across a diverse set of ecological conditions. How many researchers do you think study natural populations of house sparrows?
Okay, I think I should know this answer. It's definitely a smaller number than mice. I don't know, I'm going to say 400. And I think the point that I want to raise here is that, whatever the number is, we really need these kinds of systems to gain a better understanding of ecology, evolution, and other aspects of biology in the messy and complex natural habitat that organisms exist. Model organisms are definitely great for certain purposes, but only things like house sparrows are going to be suitable for other questions.
Totally agree. And fortunately, there are many such systems. For example, we talked with Peter and Rosemary Grant and Trevor Price about their influential work on adaptive evolution and speciation in Darwin's finches. And today's guest is David Reznick, a distinguished professor of biology at the University of California, Riverside, who's also made major contributions to our understanding of adaptation in the wild. David's name is synonymous with the Trinidadian guppy, a small freshwater fish that has made a outsized splash in our understanding of life history evolution.
Oh, groan. Art would be so proud of that terrible dad joke. But anyway, life history traits that David has studied are those traits related to how organisms grow, when they sexually mature, how they invest in reproduction, and how long they live.
The combination of traits a population has is referred to its overall life history strategy. And a fundamental question in biology is to understand how and why different life histories evolve.
For example, why do some albatrosses delay maturity until they are 10 years old, lay only one egg every other year, and then live to be 70 years old? But most songbirds become sexually mature after just a year, can lay dozens of eggs in that year, and only live a few years.
David's research has been critical in providing empirical evidence for the role of age-specific mortality in driving life history evolution using the Trinidadian guppy system.
What's great about the guppy system is that populations along the same river drainage are separated into areas where they experience either high or low rates of mortality because of getting eaten by predators in the communities.
Yeah, so in large lowland rivers, little guppies are part of diverse fish communities. And essentially, guppies are food for really big fish. This results in a really high mortality rate from predators and selects for a fast life history where guppies mature to small size and a young age, have lots of babies, but don't live for very long.
But as you move upstream into the northern range mountains of Trinidad, barrier waterfalls prevent the big predatory fish from moving up, resulting in only guppies and slightly larger killifish from occupying small tributary streams. Mortality there due to predation is much lower, and guppies have evolved a slow life history, where they mature later, have fewer larger offspring, and live longer than the lowland counterparts.
This pattern is repeated along river drainages all across the northern range mountains of Trinidad, and genetic analyses have shown each river drainage represents an independent evolutionary origin of these life history differences.
Over the years, David has treated these river drainages as replicate test tubes in nature. He has moved guppies among streams and shown that these life history traits can rapidly evolve.
These results have been highlighted in biology textbooks and are really well known. But over the last decade or so, the story about guppy life histories has itself started to evolve, as new results challenged old narratives and other processes like density dependence, in addition to mortality, were incorporated.
We talked with David about how he became a biologist, how he came to study guppies in the first place, and why he has recently become interested in eco-evolutionary dynamics in his system.
We also talk with David about a second arm of his research focus, which is on the evolution of placentation in the Poeciliid family, which includes guppies.
I'm Marty Martin.
And I'm Cameron Gallenbohr.
And this is Big Biology.
David Reznick, welcome to Big Biology.
Thank you for having me. It's an honor to be here, Cameron.
Yeah, well, so to begin, for full disclosure to our audience, I have a little bit more of a personal connection to our conversation today because you were my postdoc mentor and advisor, and we continue to collaborate. So I'll try not to share embarrassing stories about you if you don't have any embarrassing stories about me.
Okay, agreed.
So, David, I may not have the same connection as Cam, but your research has been really influential for the things that my lab does, as well as a whole generation of evolutionary ecologists. And when we get conversation started, what we like to do is some origin stories. So when did you decide to become a biologist? Was it something you wanted to do from an early age or something that you came to later in life?
I think since I was old enough to know how to walk, I was interested in biology. You know, because I grew up up until the age of 7, I lived on the shore of the Long Island Sound. And times were different then, but certainly by the age of 5, I could walk out the door and wander on my own. I don't see 5-year-olds wandering around these days, but my favorite thing to do is to go down to the beach and look for, the big treasures were the sheddings of horseshoe crabs and other kinds of animals. And I was the kind of kid who, we had an outbreak of some kind of worm in the sound there, and I collected a jar full of them and left them on the back porch where no doubt they died and smelled very badly. And there were elves in those days who would clean up after you. and so it really, in many ways, there are wrinkles between the beginning and the end, but in many ways, what I do now for a living is an extension of what I did as a hobby, beginning at a very early age.
So, David, you did your undergrad at Washington University in St. Louis, and then you went on to do a PhD at Penn. Bob Rickliffs was your PhD advisor. So let's talk a little bit about your time at Penn and sort of the state of the field during that time. So, I imagine this is like the late 70s, maybe early 80s.
No, I arrived in 1974. I'm older than you think, Cameron.
Oh, you're so young at heart. Okay. So, but kind of leading up to the early 80s, Steve Stern had published a few review papers on life history evolution. Charles Worth had his age-structured evolution, age-structured populations, I think came out maybe late 70s, early 80s.
1980.
And yeah, and so the field of, there was a sort of the beginnings of a real kind of growing field of life history evolution. So I'm curious, like at the time, as you were interested in life histories, like could you anticipate that this was like the beginning of a field of research that was going to really explode? Or was it not so obvious in the moment? Was it just something that you had a personal interest in?
I think part of it part of it was the Rickliff's influence, because the bird work was sort of on the leading edge of the empirical side of life history evolution. And so I was reading Bob's papers, and I found it very compelling. And then I was reading Don Tinkle's papers. And then a paper that people don't cite anymore was Gadgil and Bosser, which really had a lot of the critical elements. All the other people who you mentioned, those publications came out after I finished my thesis work. And so they weren't what got me going. I read them after as I was publishing my thesis work, but it was after the fact. It was reading Rickliff's and the empirical stuff. I'm much more of an empiricist at heart than a theoretician. And then it was just a couple of theoretical papers. The Gadgil and Bosser paper was a key one. The other ones in the early 70s, there were Schaefer papers that were very difficult. Then there was
Hirschfield and Tinkle, I think, had that PNAS paper.
That came out later. That was later 70s. But there's Michaud and Law. Those were the ones that but really it was Gadgil and Bosser and empirical work by Don Tinkle and Bob Rickliff's and the papers that I tracked down from Bob Rickliff's citations like Lack that got me going. And then it also was becoming, people were publishing. There was a lot of empirical work. Bianca was publishing then. And so, one of the things I did, Rickliff's had a one semester that he taught a course on, historical developments of big ideas in ecology. And one of my take-home messages from that was that by doing a detailed literature review, by asking, this is my question, when did it begin? And what have people done? And how has it moved forward? And can I use the progress made to date to define the vacancies where future work can have the biggest impact? And so it was all those things. And then the Stern synthesis actually came later. Like I said, I was pretty much done with or was I done? I was far along. The die was certainly cast by the time those papers came out. They had a big influence on me because he did such a terrific job of pulling together everything, and summarizing everything, so I did read those papers, was influenced by them.
Yeah, Rickliff's has had such a huge influence on me as well, and many other people. I gotta say, I want to talk about guppies, but let's give you an opportunity to transition to guppies by mentioning someone else that I think has had a huge influence on you in this particular space, John Endler. Did he influence your move into guppies? How did that work?
Absolutely. I mean, I can remember the day and the time and the place where.
Okay, that's specific.
So, I mean, what happened is I initially was working on mosquito fish, and I had a pretty good research program. I was making good progress. I'd done something on the genetics of offspring size that I called grandfather effects, and was doing work to look at costs of reproduction. They were turning out to be hard to breed in the lab. And the reason I was beginning to discover was because they're seasonal and you have to give them seasonal cues, which is a hard, and I was just working that way through, and I was in a critical phase. It was the beginning of my 4th year of graduate school. And I could see that I could get a thesis together, but it was going to fall short of what I wanted. And at some point, somebody told me about a talk that was being given at the Academy of Natural Sciences by some guy on guppies. And he said he thought, he knew that literature. He thought it wasn't really going to be anything original. It was just that there were a couple of guppy papers. And I don't know why. I didn't usually go. I had to bike ride through Powelton Village, which is kind of a tough neighborhood and everything. But I went. And I sat down and listened to Endler talk. And Endler was talking about the interplay between predation and female preference in shaping the evolution of life histories. He was talking about this repeated pattern of different rivers, of high predation and low predation localities, and the evolution of male coloration. And I'm sitting there and thinking, if male coloration evolved that way, it was because those localities differed in age-specific mortality risk. Because you couldn't be a brightly colored male when the predators were there, but you could be when the predators were absent, which means that you could be mature and safe. And so I went out to dinner with him and I said, this fits into life history evolution. I can predict for you what the differences in life histories of these fish are going to be. And I want this to be my dissertation project. And would you support it? And he said, yeah, that sounds good.
Wow.
So then I went back to school and I pretended I was a second year student getting ready for my oral qualifying exam. And the idea is to write a paper, a qualifying exam paper, and hand it out to my committee and ask them for an unusual thing to let me, as a fourth year student, change my dissertation topic. One of the members of my committee was a population geneticist named John Gillespie, who was a hardcore theoretician. I think he never had a high opinion of me because I wasn't theoretically inclined. And I remember him walking into the room. He was the first one to come into the room. Also, I think he wanted to fail me on my oral qualifying exam. That's a different story. In fact, I'm pretty sure he did. But yeah, he was the first one to walk into the room, and he put the paper on the table and pointed his finger at it, and he said, "This is really good." And I figure, oh, if John Gillespie likes this, I'm in good shape. And so in March of my fourth year as a graduate student, I began a new dissertation project. And I met John Endler in Trinidad. I handed him the paper when I got there. He didn't realize that I had a fully designed project. He thought I was just coming to run around and become familiar with the system. And then that night, he got out the topo sheets and the areas that he was recommending that I visit went over two different topo sheets. So I remember him aligning them and tracing them. And I thought I still had that map that he gave me, but I don't. Somewhere along the line, I lost it. And then I was there for 20 days. In 20 days, I collected about 15 different localities. I dissected. I looked at size-specific fecundity. I measured diameter of the ova. And so I had, by the time I came back on the plane, I knew the size at which females began to reproduce. I knew what their fecundity was, size specific. At that point, there weren't computers. I had a Hewlett-Packard, handheld thing that Warren Ewens had loaned to me. And I'd done calculations like analyses of covariance to show that their fecundity was higher and their babies appeared to be smaller. And I remember walking into Rick Leff's office and he's saying, "So what did you find?" And I was like, "Here it is." But the other thing was that I brought back live guppies. And, if I had done it now, the way I did it then, they would have all died. I couldn't have made it work. And yet somehow things were different then. And even though I didn't have a good background on how to keep fish, I just was lucky all the females survived. And I brought back only females because they store sperm. And so I isolated all the females and began getting the litters and creating numbered pedigrees. And then I worked from March 1978 until August 1980 with almost like 3 days off. Christmas, New Year's, every day I had to be there to get these things to work. But it was really a fantastic time because it went so well. Part of it was because they were so much easier than mosquito fish, and the mosquito fish had kind of hardened me to, how to do things right and keep the fish healthy. So John is really my surrogate advisor.
So one paper I think that, I remember reading, I guess, as an undergrad, and I know was one of the probably one of your most impactful papers was the 1990 Nature paper where, you had previously shown that there were these life history differences between high and low predation sites, but then you experimentally moved guppies from a high to a low predation environment and actually documented the rapid evolution of the life history. So can you give a little background of like how that study started and what was the motivation for experimentally moving fish from one environment to the other?
That actually, that was piggybacked on John Endler. John Endler, one of those localities was John Endler's introduction made in 1976. And I think by 1978, certainly by 1980, he had shown the evolution of male coloration. He hadn't done the background lab work, that I'd done, but yeah, it was John that did it, and also was part of a conversation with Dan Jansen. Dan Jansen was that, and Dan Jansen's famous for doing these bold experiments, and I was showing him my results and showing him the break at the waterfall on the Aripo River between high and low predation and how they were different immediately above and below that break. And Jansen said, "Well, why don't you, you could take predators and you could put them over, and so I just began to expand on the theme that John had established. And also Jansen's suggestion was that I could both increase and decrease mortality risk depending on which stream I worked in, and in a sense, treat streams as they were giant test tubes. This also, the thing I mentioned earlier, is it capitalizes on what I was looking for and what had been missing from the literature on life history evolution, which is I had replication, I had natural replication because the same community succession was present in different rivers. We now know that they represent independent origins of adaptation to the alternative environments. And I had an animal that I could work on in the lab, so I could look at the genetics of the differences among populations.
It's such an impressive system. I've been working on house sparrows for a long time, and I'm incredibly jealous of the things that you can do, from the replication to the amenability to the common garden studies and things. I've tried to do that with house sparrows, and I'll never do that again. I probably also won't do that with guppies, but
that's what I faced when I was thinking about side blotch lizards. I mean, now you actually could do that because I have the technology for breeding reptiles in captivity. The thing that I envied the reptile people for, and that I didn't think I could do, was mark release through capture, and to be able to know who's who and follow them through time. And so that became sort of the next leap for me in the 1980s when I was looking for, what the next generation of research would be. In about 1980, I laid out a research plan in my first paper that shaped what I've done for the rest of my career. But the one thing that was missing was being able to mark and recognize individuals. And so that was one of the things I began working on in the mid-1980s after I got my job.
So, David, I think one of the things that I really want to talk about, I mean, there's many things that we could cover, but I think it was 2019, you and Cam and many others wrote this paper in AmNat on what became, an example of eco-evolutionary dynamics. But before you say anything about that, can you maybe just introduce the listeners to, the kind of results of those transplant experiments, to sort of what happened to the life histories? What were the expectations? What's your interpretation of how it happens? I think that stuff is important to understand to get to those 2019 results.
Okay, there are actually many steps, so I won't give them all to you.
Okay.
I'll just
Nutshell,
abstract them. So the idea was that we knew that guppies evolve, and I thought it was because of differences in age-specific mortality, that adults have a higher risk of mortality in high predation localities than they do in low predation localities. When I learned how to do, people always would bug me and say, well, what about the birds? What about the insects? What about other things? And how do you know that the fish really is the whole answer? And that's where I began to pursue individual mark recapture, because then you can measure mortality rates. So my reaction was, the hell with all this. I'm going to measure mortality rate and find out, not worry about insects and birds and other things. I'm going to look at what's happening with guppies. And I found that the guppies from the high predation localities do, in fact, have higher mortality rates, but not in the way that they would need to explain the evolution of the life history. And that was because the increase in mortality risk was equally distributed across all size classes. And to fit in with Gadgil and Bossert, you had to have a selectively higher risk in adults and not juveniles. And in the Gadgil and Bossert model, if you change mortality rate equally across all size classes, they don't evolve. And I remember when I first got that result, I was like, it was 2 nights before I was going to go present these results at an evolution conference. And I couldn't sleep. I mean, I was walking around the streets, in the middle of the night, trying to think, how do I get out of this? And the answer was that I already know they evolve. I went in there and said, here's an odd result, but I know they evolve, and they do have higher mortality rates. That became that's when the Charlesworth book was out. And I was looking at the Charlesworth and the idea, for various reasons, I was looking at models that assumed density-independent mortality. That's what Gadgil and Bossert was. And those are easy models because the best life history is the one with the fastest rate of population growth. And density-dependent is, that's hard, because there are different combinations of things. But this and other things were pointing at density dependence being important. And so I had, the question was, how do you tell the difference? Because the end product looks the same. You can't do selection and see how they evolve, and in that way distinguish between the alternative hypotheses. And so it really was a long path to figure out how to get to that end. And so the whole point for the 2019 paper was to, before that, I had only looked at guppies 4 years after the introduction. And I thought 4 years was kind of bold. I didn't think things could happen that fast. It was like too late. And so the burning question was, what's going on during those first 4 years? And so we started a new series of experiments that was built on, it had many different components, and you can ask about those later. But it was designed to distinguish between two different hypotheses. The first hypothesis was that guppies evolve because of the change in mortality risk associated with the change in predators. The second was that guppies evolve because when you take predators out of the picture, at that point I had shown that the population goes up and that they experience density regulation. And the distinction between those two, this is something that Joe Travis and I worked out sitting down in the living room of my house in 2005, I think, or was that the timing of the change, the timing of the evolution could distinguish between the two. Because if it's predation, the most intense selection on guppies is the day you introduce them into the new environment, because that's when their phenotype is most distant from the optimal phenotype. If it's the indirect effect being mediated through density and resources, then selection may actually work in the opposite direction. They're now in a resource abundant environment because there are no guppies there. They can have great reproductive success, and it isn't until density regulation sets in that you're going to see them begin to evolve. And so that was the alternative. And the interesting thing is that Cameron's lab did the experiments, and they had long since forgotten what the alternative hypotheses were. It was like a perfect double blind. They were just every year generating the data. And I remember Cameron saying, we have these results and they're really pretty weird. We don't know what to make of them. And he sent them to me, and it was like, Cameron, go back and look at the proposal. We have a figure in there predicting what they'll look like, and you just discriminated between the alternative hypotheses. Whether or not that's eco-evo dynamics, is a different discussion, but it definitely said, and it turns out that I laid out those alternative hypotheses in my first paper in 1982. I hadn't really figured out all the wrinkles behind them, but I recognized that those were alternatives. And somebody once rejected the paper. The 2019 paper got rejected all over the place before we got it published. But one of the rejections was, hey, you said all this in 1982. And I wrote back and said, I thought proving it is different from saying it.
Yeah, that's not science, the first one. Can you define maybe what ecoevolutionary dynamics are broadly? Because I think it's going to be useful to map it back. That's a complicated study design, and it is very elegant to juxtapose those two outcomes. But I think to sort of define it, and it'll make it easier to track back to what that is.
Well, I have to first say that there are multiple definitions out there.
Of course, it's biology.
and I do not accept most of them. In fact, some of them I think really have set the field back, because eco-evo dynamics is now a different name for evolutionary ecology, the way most people are using the term. But eco-evo dynamics, we define it. There's a paper that Ron Vassar was lead author on in Ecology Letters in 2021, and that's where we sort of lay out our vision. And the vision is the same as the one that was in the Elner, Harston, Yoshida, Fussman collection of papers in the early 2000s, because theirs is the most elegant sort of theory and empirical work that does it. But the elements of eco-evo dynamics are very strong selection, coupled with having the genetic variation to sustain contemporary evolution on the same time scale of ecological change, and to also have a continuous interaction between ecology and evolution. And in the context of our paper, density dependence can be attained in different ways. But the density dependence that we were seeing and that we proved was happening was that guppies, when they get there in the absence of predators, become very abundant, and they change the structure of their ecosystem, and they impose selection on themselves. And they're evolving as they change, but also as they're evolving, they're changing their ability to utilize resources and changing further the nature of the ecosystem. Now, we've only proven really the first step. The guppies adapt to their impact on their environment. That's what the 2019 paper does. And criticism, which is a valid one, is that we haven't shown the continual interplay. That eco-evo dynamics, if you look at it in the Elner, Yoshida, Harston thing, generates cycles. And because of the cycles and the inbuilt frequency dependent selection, it'll go on forever. But I don't think that's a necessary feature of eco-evo dynamics, because if you go back to the Pimentel papers of 1963, I think they show genuine eco-evo dynamics in a host-parasite interaction, but it goes to a stable state. And the signature of dynamics is that the stable state that you see, that the ecological interaction is fundamentally different from what it would be had evolution not happened. And so the idea is strong selection, rapid evolution. The alternative, the genetic variation to do it is you go extinct, right? And then change in evolution and ecology on the same time frame, but also showing a real interaction, that one is changing the other, that there's a duality to it. And that's what Pimentel was arguing for. And it's interesting that it lay dormant. It's almost like, Mendel laying dormant for 35 years, and because it didn't really get picked up again until Elner and Harston and that research program got going. I think it's still an open story whether or not this is a prevalent feature, but that's what was new. It isn't brand new. I mean, people say, it's not new because Pimentel did it, but the point is that it isn't. Both of those were lab studies, and so the big challenge that we were trying to meet, with the research program that began in 2007, was to say, can we bring this into a complex natural community and show it in action in nature? So that's what we were trying to do.
Hi there. You've been listening to the free version of Big Biology. To hear the rest of this episode, you need to subscribe.
To do that, head to bigbiology.substack.com and sign up for a paid subscription. There are a lot of different membership options available.
Not only will you get access to the rest of this episode, you'll also get access to extras like our episode debriefs, behind-the-scene content from Keating, our artist, about how he makes the cover art for each show, and more from our guests about their path to science and their hobbies.